Up to the next level with his head.
Woo!
Good for
Excellent.
you. If I don't
All
pass
right.
out, you're gonna be fine.
Be that rich doctor.
Exactly. Always
Rich in
find
here.
out.
Rich in here.
It's the most important.
All right.
I won't be
Right.
the doctor, that's the problem. I don't pass by with him.
You won't be rich or a doctor now.
Take your pick, both or none. All right, so today is integration of metabolism. And that basically means all the pathways we've talked about, glycolysis, TCA, beta oxidation, ketone genesis, etc. All of those are happening in the same cell. How do they talk to each other? How are they influenced by each other? And especially, what I want you to think about is what are their goals, and that'll tell you what hormone drives them. So today we have the insulin guys and the glucagon guys. And that's really how you kind of divide it up. And what state do we have insulin? Do we have high or low blood sugar? Insulin is going to come out when you have high blood sugar. Great. And that means glucagon comes out when the gluc is gone. Yes.
Oh.
All right. So let's start with the insulin guys. We are fed state, and therefore insulin is out. So we're just going to put fed and slash insulin. All right. So we have nice bloodstream here with glucose loaded up. Oh my gosh, stop eating ice cream. Stop doing. Carton's gone. All right. So, and we're talking about a liver cell in particular because the liver does everything. Other cells would do a few of these pathways. The liver is going to do like all of them. All right, so the first pathway, taking glucose in, this is going to be glycolysis. And I'm writing glucose, and then I will name the pathways and underline them in, I'll write names in, let's see, orange is not going to show up on that thing. It's not going to show up anyway, so. All right, so we're doing purple for glycolysis here. All right. Glycolysis. Okay, so there's glycolysis. And this is taking glucose, breaking it down into pyruvate. And then let's go ahead and get a mitochondria. All right, we'll go ahead and draw a mitochondria like so. This is the mitochondria. All right, pyruvate, and then once we are inside, we are going to make that into acetyl-CoA. Okay? And I often just show acetyl-CoA as two carbons next to each other because that's how many carbons are in acetyl-CoA. So it works. All right, now off of the glycolysis pathway here, we have two pathways that come off at glucose 6-phosphate. What pathways branch off there? PPP. All right, we have the pentose phosphate pathway. Excellent. And on the other side, what's another one? Also coming off at glucose 6-phosphate.
Fatty acid synthesis?
That is a good guess, but in this case, at glucose 6-phosphate, we actually have glycogen synthesis or glycogenesis. I'm just going to stick a period there. All right. So if we had a lot of glucose, then we're going to be running this pathway. Think of glucose as water pressure in this case. If you have a freaking ton of water up here, a lot of pressure, then it's going to go down this hose and it's going to squirt those ways, that way. This is a more everyday way of thinking about the pathways as pressure. Another thing you could do is if you really like the reactants to products, you have a ton of reactants, you make a lot of products. That's saying the exact same thing, but some people vindictively hate chemistry. And so we're giving another option to escape that.
So
All right.
So the glucose 6-phosphate is that the hexokinase that turns the glucose into the
Glucose 6-phosphate.
6-phosphate?
Exactly.
Okay.
Yep. I'm bypassing enzymes and
No, I just
zoomed
wanted to
in.
make sure in my mind that I was at the right spot.
Yes, you are. And both of these come off of glucose 6-phosphate, glycogen, and pentose phosphate pathway.
Awesome.
All right. And I'm going to add another little one that you guys haven't done the full pathway for yet, but you have touched one molecule. What are the two products that we care about for the pentose phosphate pathway? We do it for NADPH and...
The ribose
Yes.
5-phosphate?
Exactly, ribose 5-phosphate. And that is going to give us purines. And so another pathway you're going to learn is Peorians synthesis. I don't think you've had it yet, but purine synth is also one. Look at the water pressure here. If you had a lot of glucose, would you have purine synthesis going on?
Oh, if you had a lot?
Yeah.
Yeah.
The pressure says yes. Now, these are regulated so that they can be turned off, but in general, if you have a lot of glucose, you could overwhelm the regulation, which happens in some diseases. So this one fits more into the insulin pathway than the glucagon pathways. So that's kind of a side note. All right, coming down. Once we get into the mitochondria, what's the path? What's this? What's this pathway?
TCA.
TCA. Excellent. All right.
Circle.
The TCA, yes, the circle. All right. So that's going to happen, and coming off of the TCA, we get a whole lot of NADH and etc. and FADH2, and these two guys are going to go to what?
Electron transport chain.
Electron transport chain. Excellent. All right. And I've started writing those in the wrong color again, so TCA, ETC. All right. So, acetyl-CoA. We've talked about a lot of pathways that touch on acetyl-CoA. What are some of the pathways that acetyl-CoA comes from? Sydney.
Fatty acids.
Fatty acid synthesis. Excellent. All right, so acetyl-CoA plus some TCA cycle intermediate, oxaloacetate, is going to come out and send it to fatty acid synthesis. And, I'm going to shrink my mitochondria here so we have a little bit more room.
So the fatty acid synthesis does not occur in the mitochondria.
No.
It occurs in the liver.
It does occur in the liver, but not inside the liver's mitochondria.
Okay, so the cytosol.
Yes, exactly. And so out here we have fatty acid synthesis. And that is where we have that big dimer enzyme where it's fatty acid synthase, and there's two of them put together. Yeah, sort of like that. And they have little grabby bits.
Yeah.
Remember the grabby bits? And they grab the malonyl-CoA, and then they put it down on the other arm, and they have the catcher and the holder and the catcher, and Matt's in there, because Matt is my favorite uncle. Yeah. So that's what's going on here, and that eventually gives us a, this comes out as, a fatty acid. Okay? And so that squiggle is just a fatty acid because a fatty acid is just a long chain of carbons, okay? And coming from there, we've done fatty acid synthesis. Now let's do, what do we do with those fatty acids? Do you guys remember?
Triglycerols.
Tags, triacylglycerols, exactly. And so now you have tag synthesis. All right, and tag synth, you are going to take a glycerol backbone and stick those acyl groups, which is what this is, acyl, onto them, and you do the first two, and then the third one you have to get rid of that phosphate off the glycerol backbone, remember? Yeah. Now, when we're backed in a corner, literally. Oh my gosh. All right. But right here, we can split into either finishing tag synthesis, or we can keep the phosphate and turn them into special sphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphing phospholipids. Excellent. And so you can take this, which is called a, what do you call a glycerol with 2 acyl groups and a phosphate? That is what they should have called it. But it is called a phosphatidate. Phosphatidate. And that's why we have phosphatidic blah, blah, blah. Phosphatidylcholine, phosphatidylserine, phosphatidyl whatever the heck. And that's because it's using a phosphatidate plus whatever we stick onto the phosphate group. So over here, if we keep the phosphate group, then we will keep phosphate. Then we get phospholipid synth. All right. Whereas, if we don't, then we get a tag. And the tag is going to be stuffed into a transporter called VLDL. VLDL. So you could say lipoprotein synthesis, but we didn't really go into that as a, how are they put together? You stuff stuff inside of them and you kick them out. Good job. All right. So that's one thing coming off the acetyl-CoA, fatty acid synthesis. Then what you do with the fatty acids, make tags, make phospholipids, and then ship them out. Okay? Now, I would like to back up a little bit. All of the reason for this is that we have a lot of glucose that we want to not waste. The body hates wasting stuff. Even if you are diabetic and have a blood glucose that is like 4 times normal, your kidneys are going to be trying their very hardest to bring all the glucose back, even though it's literally, you know, wearing your vessels out, giving you inflammation issues, and it will eventually cause severe health issues. But the kidney still doesn't even have a mechanism to say, you know what, we have enough glucose, let's let it go. So, in like fashion, any glucose that goes into the blood, the liver is going to be like, okay, let's use what we need and then store the rest. So using what we need to make products, that's going to be like glycolysis and TCA to make your ATP out of the electron transport chain, and it's like, energy. Now that we've got all the energy we need, where are the places that we store it? What are some storage forms of glucose? Adipose. That's a big one, and that's what all this is. We have lots of glucose. We want to keep that energy somewhere in the fat. What is another? Muscle is... Oh, glycogen is... Yes, so muscle holding glycogen. I see what you're saying. Yes, in that case, muscle will hold glycogen, and glycogen is another storage form. The liver will also hold glycogen. And so, but once glycogen is full, then
it's all out of those
fat. And so you make a freaking ton of fat. All right? So, hopefully that makes like clear sense. You pour glucose in this side, it has to come out somewhere. It's either energy, fat, or glycogen, because you're going to keep it. It's not disappearing. All right? So, what are some other things we can make? Because all of this is a making experience from acetyl-CoA on. When you have excess glucose, you're trying to make things with it because you have it. So what are other things you can make from acetyl-CoA? Cholesterol. Excellent. All right. So, I'm going to get rid of that right there. All right. And my mitochondria is way too big. I swear, we need to like cram into a small study room. I draw so much better on those kind of boards. All right. Tell myself that. So, cholesterol synthesis. All right, so we have cholesterol synthesis, and that kind of covers all the ones where we are trying to use glucose to make really, to make things or to store it. All right, and did you have a question?
You said it's used for energy storage, and what's the other one?
Energy storage, or you're either going to, there are three things I guess you could say. Glucose is going to be used for either energy, like ATP,
Yeah.
or making special products,
Okay.
or store it. It will not be wasted, though, is the point.
Okay.
Should one of these pathways coming off from the cyclical may be glycogen synthesis?
Glycogen synthesis?
Is it?
Glycogen synthesis is one of the pathways, you're right, and it's up here off of glycolysis, off of glucose 6-phosphate.
Oh, so it comes all the way. Okay.
So when you're saying put special products, so you're talking about the cholesterol, the ribose?
Yeah,
Okay.
cholesterol, purines.
What about amino acids up there in that square?
Okay, so amino acids are typically going, you might do amino acid synthesis,
I
but
guess off the pyruvate.
off the pyruvate, you would have some off the pyruvate. It depends on what their carbon structure is. You remember how some are pyruvate, some are TCA intermediates, and if you were going to make those, then you might use the energy to do it, but it's sort of a loose connection to glucose in that case. You don't just spontaneously start making protein when you don't need it. So usually proteins are going to be changed into each other. If you do get to the point where you need proteins, then you will start using glucose to make protein. But it's not like oh, you have insulin? We're going to make protein. Because you don't have anywhere to store the protein. So it's an as needed. But yeah, that is connected. I'll make a note up there. Question?
So yeah, the question, so when she has this, doing this, so in the fed state, we're getting essential amino acids into our diet.
Mm-hmm.
So is there anything here that is going to attach their metabolism with what we have right here?
Okay, how do we attach proteins to metabolism of glucose? So, that's the question, right?
Yeah.
So, the way that I would do that is I get another color, because that gets... All right, so normally I do proteins on a different one, but I'll go ahead because then it gets like a ton of lines.
Like a sketch. You don't have to
Right
go into
here,
detail. So the pyruvate
and then all through here, and then some can come out through here. So TCA cycle, acetyl-CoA, and pyruvate. If you remember, those are where the carbon skeletons end up, are in those three places. And so you can go in backwards with some of those.
Okay.
And so then you would make, we're going to take succinyl-CoA and break it back down into MTIV, methionine, threonine, isoleucine, and valine. So, question?
I apologize if I missed this. So phospholipid synthesis is part of a fed state system?
Yes.
Okay.
So you have a bunch of glucose, you use what you need right now, and then you store the rest as fat. And so...
But protein synthesis, or not synthesis, but protein metabolism, is that not considered a fed state process?
If you have enough energy, why would you want to break down protein for energy? Right now we are in a high energy state. Glucose is free for everybody. Everybody in the body has enough glucose. No reason to break down protein.
So does that mean that it
Or
just
fat.
gets formed into fat and stored?
The glucose or the protein?
Well, so if the protein comes in and you're not utilizing it,
Yeah.
what happens to it?
Then you would turn that protein into pyruvate, acetyl-CoA, and TCA cycle intermediates and break it down for energy, because you don't just, I ate a lot of protein, therefore I make a lot of muscle. That would be really nice until it was really not nice.
What about?
So yeah, you would break down extra protein.
But if you're a growing human, like if you're a kid, then you'll take some of your protein and turn it into protein.
Yeah, you will go and integrate it into muscles and other. Every cell needs protein to do its make its machines. It's little proteins that do everything. So you use the protein you need, and you don't, and then you break down all the extra.
Would you use amino acids though in the fed state to have your proteins for later, just to build your, not break down the proteins, but just to build them for later? Do you know? Or
So if you don't need them right then, you break them down, because
no?
there's nowhere to
Okay,
stick them.
Never mind.
I mean,
Yeah, yeah, that's right.
Yep.
Thank you.
and if you just made extra muscle, just think about it for a sec. Muscle takes energy to maintain. And so if you made a freaking ton of muscle, just because you ate a lot of protein, and you were, it would be, number one, we'd be huge, but number two, we would be burning so much energy that we'd have to eat a lot more, and basically as we got bigger and bigger muscle-wise, we'd have to eat more and more, and eventually we might not be able to keep up in most non-American societies, like past years, I should say. Eat less meat, yeah. That would be a good
That's
connection.
like her. She eats and eats and eats and doesn't do anything.
Some of us just have that problem anyway. It doesn't matter how much you eat. It goes somewhere. That's like magic right there. That one's magic. When glucose doesn't go anywhere, it just disappears. And then you're still tired, and it's like, what? Okay. All right, so that is the insulin fed state pathway kind of integration. Next, we have fasting, and the hormone of choice will be fasting. Glucagon, because the gluc is gone. All right. So, in this one, I am going to go ahead and draw a fat cell over here. Bloodstream right here. And fat is going to go ahead and throw some fatty acids out, and then they will attach to albumin just randomly, and albumin will take them to the liver. That's not my growth, this is albumin. Albumin.
Just the chain, right, Joe? Just the chain of fatty acid, not the whole.
Just the acyl group, yes, exactly. So you take tag, break it down into an acyl group, and then you throw it onto albumin. Albumin brings it to the liver. So this is liver. Hello, liver. Okay. So the liver is going to have the fatty acids come in, and we have a mitochondria here, and the acyl group is going to get its CoA, and then it will be acyl CoA, and dies inside. Now inside the mitochondria, what happens to the fatty acid? How do we, we're trying to get energy, how do we break a fatty acid down to make it into energy? Because we are in a fasting state, we have no blood glucose, we have very low blood fuel. Yes, what's that pathway called?
Beta oxidation?
Bam, beta oxidation. Beta ox. All right, beta oxidation does that oxidation, dehydration, or reduction, or whatever it is. Anyway, I'll pull up details if we want to go into it. But then it goes through its little 4-step process and ends up as acetyl-CoA. Okay?
So now the green circle is our mitochondria.
Exactly.
So, are you trying the product of beta oxidation to acetyl-CoA?
Yeah, so the lipid is going to be cut up into acetyl-CoA. And that's what beta oxidation is. It's just cut up the fatty acid pathway. except we call it beta oxidation because that sounds way like higher pay grade, and making money is good.
So you take one two squigglies is a CoA, and it
So-
gets beta oxidized into one.
So this is the same squiggly. It came in as an acyl group. Acyl. Sorry, I'm skipping a lot of steps because it's going for concept, but it is an acyl group, and then we add a CoA to the acyl group. The reason I bothered writing that is to remind you something does happen in the cytosol before the actual cutting it up.
That's when the carotene,
Yeah, the carnitine, excellent.
comes in and goes. And
Carnitine shuttle is right
then
here.
the shuttle.
So we have the carnitine shuttle that it's going through.
Yeah.
Carnitine acyl transferase 1 and 2.
Cat 1, cat 2.
Yep, cat 1, cat 2.
No, I forgot what's that reaction?
That's the beta oxidation.
No, in the cytosol. It's different.
In the cytosol, so we have acyl CoA synthetase taking the acyl group and then sticking a CoA on it. The reason it does that is because then the liver knows for sure, it turns on the enzyme acyl CoA synthetase when it wants all fat in it to be broken down into energy.
Is there a name for that reaction?
Nope. I mean, it's acyl CoA synthetase. It's part of beta oxidation, or I should say, it's part of fatty acid breakdown. The part that we usually zoom in on is beta oxidation, which is this right here. That's a good point. I'll write down fatty acid catabolism. Catabolism is build up or breakdown? Breakdown. Anabolism is build up or breakdown? Build up. Metabolism means whatever. Yeah, up or down, we don't care.
That's when you don't know to use that word.
Yeah, metabolism is when we don't know what you're talking about. Okay, I like it. All right, so now that we got acetyl-CoA, we are in a fasting state, so what are we going to want to do with this? In a fasting state, what's our blood glucose?
When's that thing? Under beta acid.
Oh, sorry, that's 4 steps. I'll just get rid of it. It's messy.
Oh, and that's oxidation reduction and oxidation hydration
Yes.
thing.
Yes.
That's where
Okay,
you go
I
to
like
gluconeogenesis.
Gluconeogenesis would be from here, yes. So we are going to do gluconeogenesis. Gluconeogenesis. All right, and we're desperate to get food into the bloodstream in fasting and starvation state, which is what we're talking about here. What is our backup kind of food? Because this is really slow. Gluconeogenesis, super slow. Glycolysis, excellent. That is much faster, but it runs out really fast. But that is one. So we have glycolysis. Glycogen lysis. Glycogen lysis. Yeah.
Analysis.
Glycogenolysis. All right. I memorized those both, glycogenesis and however you're supposed to say that one with an extra syllable, and now it's for life. I'm just going to write a textbook and make it right. Okay. So, yeah, all right. So those guys are going to give us glucose. Excellent. Now, when we run out of glycogen and our glucose, we get a little bit of glucose. When we run out of glycogen, and so this no longer works, and this is too slow to carry the whole body on its own, the liver just making new glucose because this is slow. And so we have to have other things that we can do. We will make enough glucose for red blood cells, excellent, and maybe the brain. Okay, good. So glucose will go to red blood cells and secondarily the brain. Now, if we run low enough on glucose, then the brain doesn't get any. What does the brain get instead? Ketone bodies. And so ketone bodies are going to be made out of what molecule? Yes, and that comes from two acetyl-CoAs being put together. And so acetoacetate, CoA, and then goes through its pathway, and we have ketone body synth. Colors. Ketone. All right, and the ketone bodies will go up into the blood, and then we will have ketones that have a double bond oxygen somewhere in them, because that's what a ketone means, and those are going to feed rest of body. And that may include the brain in starvation. In starvation, that's what switches between fasting and starvation. Besides that, it's pretty much all the same. Basically, we have enough glucose for the brain in fasting. In starvation, we run out, and it's like, okay, brain, you get ketone bodies, and that's it. And
so
then
in our, do we make our acetoacyl thing in our mito? Or is it out of our mito?
I believe it is done outside. Double check it, but I'm like 80% that it's outside in the cytosol. So acetoacetate. All right. Good deal. It's Pedro approved. All right, and so we will also have some TCA cycle going because we still need energy. And we will have the nice little proteins. I'm going to stick the same little stars. Just think about the proteins. It's much easier to just put stars than write out entire pathways for proteins, because if we're really desperate, we're going to start breaking down anything we can. And that means we make glucose to use it, of course, and ketone bodies, but also proteins is what we make the new glucose out of a lot of times. Because pyruvate right here, pyruvate, we get that from protein skeletons.
Pyruvate is the intermediate from acetyl-CoA to gluconeogenesis.
Yes. So there's like this, except acetyl-CoA can't magically become. So you guys remember how acetyl-CoA can't be used for gluconeogenesis?
Right.
Would you like me to explain that, or does it make sense? Explain it. Okay. So acetyl-CoA is a 2-carbon molecule. Pyruvate is a 3-carbon molecule. And so we would have to get another carbon from somewhere. Okay, great. Well, acetyl-CoA could go through the TCA cycle in which it joins oxaloacetate. 2 + 4 = 6. Great. Let's just take that 6-carbon molecule, cut it in half, and make it pyruvate through some enzymes. We can do that, but then we'd run out of oxaloacetate. And if we use up all the oxaloacetate that way, then acetyl-CoA can no longer even go through the TCA cycle because there's nothing for it to join. The oxaloacetate is gone. That means you broke your TCA cycle. Because there's oxaloacetate is supposed to be the 4-carbon molecule right here, and you have to have it join the 2-carbon molecule, and then you have all those intermediates that you loved learning about.
What about the odd chain? Can it use the carnitine?
The odd chain fatty acid, you are absolutely right. There is a tiny bit from beta oxidation that will make its way up to pyruvate. So you can use that one last bit.
Is that the propanol?
Yeah.
Yes, propionyl CoA. You can use that little bit to make 1/2 of to make a pyruvate, basically, which can be used to make glucose. However, acetyl-CoA cannot be used to make glucose, otherwise you would break your TCA cycle because you would rob it of all its pieces. And so these are supposed to be recycled pieces where you use them again and again. And if you run out of oxaloacetate,
Okay, so after beta oxidation, it goes to acetyl-CoA, and then it was safe to go into gluconeogenesis.
so beta oxidation, perhaps I should have used 2 different colors. The fatty acid catabolism usually is going to go beta oxidation, acetyl-CoA, ketones. So fats go to ketones because they all end up at the acetyl-CoA. Proteins and the tiny propionyl-CoA go to gluconeogenesis.
Oh, so there's no connection between gluconeogenesis and acetyl-CoA. There's
Correct.
yeah. Okay.
Very, very few. There's not really any. So I'll go ahead and like that is going like this. Okay, so the orange highlighted bits are the pathway of fat, and then the green highlighted bits are going to be driven by, largely by protein. And some of the proteins can make new intermediates in the TCA cycle. Remember how they can become succinyl-CoA, oxaloacetate, and be broken down into those? Those extra ones, we can take them and break them apart without any problem. And so those protein made extra spare bits, so the extra oxaloacetate, the extra succinyl-CoA, those can be used to make new glucose as well.
Would the orange highlighted line, would it go around this TCA thing?
Yeah, actually, you're right. You can use it. You can use acetyl-CoA to go through the TCA, definitely.
All right, good. No problem.
Awesome. And that is one thing I remember. I was sitting in a TA session where Sweekar was teaching, and he was like, for some reason, you can't use acetyl-CoA, and it was really complicated, and basically, the bottom line is, just remember you can use everything but acetyl-CoA. So if you like it that way, go for it. You don't need to remember necessarily why, but if you can, it's cool.
What was the green one for again?
The green is for proteins. Proteins.
And it goes from, it starts from where?
Proteins, if you remember the protein carbon skeletons, wherever those, so these 3 stars, those carbon skeletons can be broken down into pyruvate, acetyl-CoA, or TCA intermediates. You remember how like M3IV, all of those, methionine, M3 threonine, isoleucine, and valine, all become succinyl-CoA. Well, those are in the TCA cycle. And then we can take those extra TCA cycle bits and use them to make glucose because they're extra. If they weren't extra, we wouldn't use them. Proteins make extra ones. And so then that's gluconeogenesis. And clear up here, we have something that doesn't come from fat. It doesn't come from protein. It's all on its own. That's why it's so nice. And it's just glycogen. It's basically glucose that's ready to be cut straight into glucose. So that's why there's no underlining there, because it is basically glucose. Cool? All right. So last thing to say is that, no, actually I think that kind of covers it. So
I have a question. I don't know if you're going to cover it. If you do, just tell me. So what is the difference between a fasting state and a starvation state?
okay, good question.
Other than glycolysis or glycogenolysis is gone.
So that kind of almost is the, so no, okay. Fasting is going to last roughly 3 days. Glycogen will be gone in the first day. 1 day, glycogen's gone. 24 hours, generally. Then you have 2 more days of fasting, and starvation starts after 3 days. And that's when you start breaking down more proteins and getting, you're taking energy sources that are more damaging for your body to access. So you're taking proteins, and if you, yeah, so it's basically 3 days, and then you move into starvation. What is the exact biochemical, this is fasting, this is starvation? I've always understood that it's when it starts taking proteins more heavily, and you're relying on ketones, and then long-term starvation, you go to relying directly on fat.
So the fat, so basically it's fatty acid, metabolism, and the breakdown of amino acids. Those are the two things for starvation.
Fatty acid and, say that one more time, sorry?
Fatty acid and amino acids?
Yeah. Yep. And so basically, it's when you've gone long enough without glucose, you'll use everything else. Because ketone bodies will be there too.
So biochemically, are they truly saying that starvation is when the fat source has been utilized and you're breaking down more of the protein?
Let me draw a timeline.
Okay.
So, all right, timeline.
Tell me that urea cycle is going on in starvation.
Yes, urea cycle will be going on in starvation because you've got all that nitrogen to take care of when you're. All right, so we have, let's see, at the beginning here we have eating. Eat. That means fed state. All right, and so that lasts 2 to 4 hours. And your glucose, which I'm going to do in blue, is high. However, it quickly starts to drop. And by 24 hours, I should say that it starts to drop. All right, it is relatively quick. But after 24 hours, you, after the 4 hours, you're considered beginning. Okay, 2 to 4 hours is fed state. After that, you start moving into early fasting. That will last up from 24 hours is a marker for when glycogen is gone. Glycogen gone. And 3 days is when you move from fasting to starvation. And at that point, you're going to be using more amino acids and ketone bodies. And after 2 weeks, you are going to, my glucose thing is not great. There's a better one in your slides, I think. But after 2 weeks, you are going to be using more fatty acids directly. And that will continue on. So between fasting and starvation, you are going to use ketone bodies in both and amino acids in both, but it gets more severe in starvation. So, and that's the best I can give you. It's kind of a hazy differentiation between the two, but yeah. So I like to remember this as there's 2, 4, as in 2 through 4, and then 24, so it's 2, 4, 2, 4. 2 to 4 hours for fed, 24 for glycogen being gone, and then after that you have 3 days, and I'm going to double check that. I think it might be 3 weeks rather than 2 weeks.
What does it say on the very top, right above?
Eat. So at the beginning you ate. So it says eat and fed.
Oh, okay.
Because I was writing in my language rather than English. Eat, fed, monosyllabic words. All right. So the idea behind this, hopefully with you could make like a super rudimentary sketch of this where you just draw glycogen as an arrow. I'm going to go ahead and do it on this other one. But travel time. Oh my gosh, you're flying. All right. So the way that I would check to see if I really have this and to review this is I would say Insulin pathways, go. All right, I know that's glycolysis. I know that's glycogenogenesis. I know that's PPP. I know that's TCA. And then I know that this is fatty acid synthesis. And then I know that from fatty acid synthesis, I get two things. I get tags, and I get phospholipids. Is there anything else? That's pretty good. All of that is driven insulin. And then I would just, frankly, throw in, all right, now for glucagon, which is fasting and starvation. Right here we have beta oxidation, and then everything else just goes backwards. And that's how I kind of review, and then I ask myself questions. What would happen if, which pathways would be going if, and I come up with scenarios such as, I'm diabetic. My glucagon is up, but I have a freaking ton of glucose in my bloodstream because I'm type 1 diabetic, and I can't make insulin. And so my glucagon is always high because insulin isn't, you know, they're not brothers where it's like insulin's beating glucagon down so they're all on par. Type 1, glucagon's always high, which is their problem. And insulin is always gone. So they have high blood glucose, but they have a lot of glucagon, which isn't usually paired together. What would happen? And actually, let's do this one, because this one's actually probably one of the more interesting ones.
Glucagon is high,
Glucagon is high.
so that means that you're starting gluconeogenesis.
Yes, and that,
And that's going to lead to ketoacidosis. Well, it's going to lead to metabolic acidosis because the blood sugar is going to go so high that the muscles, that everything, the cells can't take it in. Because without the insulin, the blood sugar is going to go high, but the cells don't get it.
yeah, so glucagon will make gluconeogenesis. It will also do ketogenesis, because glucagon, really quick, the really easy way to think about what glucagon does is bring any food into the blood. I don't care if it's glucose, it's what it's named after, but I don't care if it's glucose, I don't care if it's lipids, I don't care if it's ketones, bring all that food into the blood. And so if you have high glucagon, that's what drives all the regulation. So you're going to have gluconeogenesis, you're going to have ketonegenesis, and that's where the ketoacidosis comes from. And then you're going to have all that in the blood. But with no insulin, it can't take it into the cells very well, so it just stays in the blood. And so just, yeah, have fun thinking about different things. What if this pathway is broken and you have insulin? What would happen? Think about stuff like that.
G6P, hold up.
Yes, these two would go, those would go out the wazoo because there's nowhere else for them to go. And so you would have a ton of glycogen, and there are some glycogen storage diseases where you get too much glycogen. And PPP would be going, which means you might end up with gout because one of the, you guys, I was going to ask, but you haven't had the pathway yet, but Peorians, when you have a bunch of Peorians, you can make uric acid. Anyway, but yeah, so you'd make a lot of PPP products. You'd have a lot of NADPH, and you'd have a lot of Peorians. So what would happen if we broke it here? And it was fixed right here. Fatty acid synthesis doesn't work very well.
Deplete your lipids, you'd have a low lipid.
You would have low lipids. You wouldn't have special or non-special. I'm so sorry you're not special. You wouldn't have lipid products, and that would be, I don't even know if you would survive with that kind of a defect. So yeah, that's kind of what I would do to see if I have this down and understand it. So, any other questions?
Is that a down arrow? Is that
It's the phospholipid.
It's PL, phospholipid.
Oh.
I was doing chicken scratch over here. Now you know what chicken scratch really looks like. All right. Yeah, hieroglyphics, I'm telling you. Great. All right, so I'll go ahead and turn this off. Have a good one.